Another report, Bighorn sheep disease uptake, Peri, Wolff.
It's loading my presentation here. Are you the expert? Can we cover that off?
They've been doing some research after this at Cabela's. Besides, you're making me work tomorrow. I should make your favorite time of the week.
Sheep, you really charges
No,
too.
unclassified. Doing a good job.
Okay,
Go ahead.
this has got a kind of technical. We turned the lights off, so it's after lunch. I apologize. I'll try to make some of the monotonous words hopefully mean something. So going to give everybody an update on where we are, and also go through a little bit of history and kind of take you through some of what the science is showing us and how the disease and the bugs act, because I think that in order to really understand what goes on with bighorn sheep and respiratory disease, it's important to kind of have an idea of how that all works. If you haven't heard, we're not unique. There are 9 individual die-offs occurring this winter in 5 western states. So far, there's been plus or minus 900 sheep mortalities across the West that have been reported. In Nevada, we've lost 103 out of an estimated 180 of our East Humboldt herd, and then we've lost plus or minus 34 out of an estimated population of 160 in the Ruby Mountain herd. As I covered last time, our clinical findings pretty much have been the same. Coughing sheep, nasal discharge, mostly not really a snotty nose, more kind of a cloudy to slightly snotty nose. The sheep are lethargic. We've seen some weight loss, but not always consistent weight loss. That's something that's a little bit different than last time I updated you. And then, of course, we've had a tremendous number of deaths. Again, the main finding that we're seeing is this in the right lung lobe, and I think that might be is this a pointer? I don't know if that's a pointer. Okay, I'll just use this. In the right lung lobe here, this is the head of the sheep up here and the tail of the sheep back there. So what you're seeing is this fibrinonecrotic bronchopneumonia, most commonly in the right cranial lung lobe, but in more severe cases of disease, you would see that extending through the majority of the lungs. We had one ewe that died where only about 25% of her lung was still functional, and that was just these little tiny strips back up here against her backbone, and the rest of her lung was pretty much affected. So I'm just going to give a little overview of history of pneumonia in bighorn sheep. Back in the time, early 1800s, there were predicted to be more than 2 million bighorn sheep on the landscape in North America. There are reports from the mid-1800s where they were starting to see that there might have been some detrimental situations when bighorn sheep started interacting with domestic sheep. They found that bighorn sheep populations which shared habitats with domestic sheep were failing to expand. And then what really kind of sort of hit it home is in the 1980s there was some accidental grazing of domestic sheep next to an enclosure that had some bighorn sheep, and all the bighorn sheep in the enclosure died of pneumonia. Certainly what we've seen in modeling and from historical records of what's been happened in Nevada is that in the 1860s we had a lot of sheep on the landscape. By 1960 there were few, And then as we've done augmentations, certainly those numbers have increased. And we know that disease does play a big role, but obviously that's not the only thing. Habitat competition, more humans on the landscape, all those sorts of wildfires have obviously played a role too. So what's been sort of recognized as the problem is that when bighorn sheep commingle with domestic sheep, it often results in an all-age die-off from pneumonia. And then almost more devastating than that is that the lamb recruitment in subsequent years is poor. And by lamb recruitment, I mean they usually see lambs that are born, but then at about 1 to 3 months of age, many of them will get sick and die. So that's usually what's been seen with these die-offs throughout the West. So what are some of the complicating factors and what's led to, I think, a lot of the controversy and misunderstandings that have been going on and on for years and years around this pneumonia issue with bighorn sheep? One is that, and this is common also in domestic animals and other species, because all ruminants, anything that's got 2 little hooves, gets pneumonia. That's one of their big diseases that they get. And oftentimes, whether you're talking about a cow, a sheep, a deer, a bighorn sheep, you can get a mix of different bacteria that you grow out of the lung. So you usually just don't see one bacteria or organism that's in there. So that's what they found in a lot of these bighorn sheep die-offs. We know that bighorn sheep can't exist in certain situations next to domestic sheep, and there does not appear to be a die-off. So what's that all about? Bighorn sheep have experienced die-offs with no known prior contact with domestic sheep. Certainly, I'm sure that bighorn sheep were dying of pneumonia long before domestic sheep got on the scene, but maybe not to the extent that they have since domestics came to North America. And then also, we're culturing some of the pathogenic bacteria out of the lungs from apparently healthy bighorn sheep. So those are some of the complicating factors that have made this a little bit of a contentious and poorly understood issue. Over the past, I'd say, year and a half, there's been a tremendous amount of science that I want to go over with everyone that really, I think, helps, for me at least, helps clear things up and has made me understand this whole issue a little bit better. So what does the science tell us? The primary players, and again, this is not unique to bighorn sheep. This is if you're a cattle rancher, if you're a domestic sheep farmer, you're going to know that Pasteurella pneumonia is a huge issue in ruminant species. There's billions of dollars spent by drug companies on fighting these 2 bacteria. The big names that you hear, and part of the problem with a lot of this is they keep changing the names. As the scientists start teasing these bacteria apart, they then rename them after themselves. So the one that probably the most benign is Pasteurella or Biberstenia trehallosi. We're just going to call that P. trehallosi. The one that everybody hears about that's usually the bad guy in the group is the Mannheimia hemolytica. So hemolytica means that it is breaking down red blood cells and does that on these culture plates, and that's one of the ways they can identify it. And then the other one that's also found in a lot of the lungs of these animals and is common is Pasteurella multocida. So these are the 3 that you're going to hear about over and over again when they're talking about pneumonia, whether again you're talking about bighorn sheep, cattle, or domestic sheep. So what makes these organisms so deadly, whether you're talking about, cattle, sheep, or bighorn sheep? They produce something called leukotoxins, and it's basically their big machine gun that they pull out and blast away, and that's what enables them to cause a lot of damage in the lung and kill the animal, get their job done. It's a toxin, and it enhances the pathogenicity, enhances the badness of each of these organisms. The other unfortunate thing about it is it attacks the white blood cells, so kind of lures all the white blood cells into the lung, and then when they all get there, the leukotoxins go in and kill them, so they can't effectively fight off the infection, and it allows the bacteria to take hold. And the bad thing for ruminants is these bacteria, because they're so common in ruminant pneumonia, they have a real affinity and draw in these white blood cells from ruminants, and that's what makes these pneumonias so deadly in so many of these different animals. So they lead to death of the cells and cause a tremendous amount of tissue damage, which usually ends up causing the animals to die. And the other unfortunate situation is that bighorn sheep appear to be rather wimpy in with their white blood cells to these leukotoxins. For some reason, they never evolved on the North American continent to deal with a lot of the leukotoxins that they have met since some of the domestic animals have come on over. So they're just unprepared and have not evolved to handle a lot of these. So what do we know about the ones that we see most commonly in bighorn sheep? And that's M. hemolytica, the bad guy, and Trehalose, who's kind of the nicer of the two. So they can have varying levels of leukotoxin, and this is where it gets complicated, is that not all of these are created equal. Some of the studies that they have done is where they've taken Mannheimia hemolytica with high, they can go in and test for, they carry leukotoxin genes, and they can also tell whether the leukotoxins are bad, deadly, or not so deadly. So they've gone into healthy domestic sheep, and they've been able to take out Mannheimia hemolytica that they know has high leukotoxin activity. And they put that in a bighorn sheep, and the bighorn sheep is dead in 48 hours. So obviously, bighorn sheep just don't handle this very well. When we look at healthy animals, go out in a field of healthy domestic sheep, go out in a field of healthy bighorn sheep that are fairly isolated. They've looked at a lot of the thin-horn sheep up in Alaska that have absolutely no contact with domestic sheep, so they're pretty pure populations. When they go in and culture them, what they find is that in the bighorn sheep, they mostly culture the Trehalose, and greater than 85% of what they culture is leukotoxin negative, meaning it's not expressing any type of leukotoxin. And then when they do culture if they do encounter leukotoxin activity and they do counter M. hemolytica, it usually has very low leukotoxin activity, which would make sense to me because it's obvious that these bighorn sheep can't handle high leukotoxin activity. It would kill them. So it makes sense that normally they have to carry bacteria with lower leukotoxin activity because they can't handle it any other way. When you look at domestic sheep, and again, these are healthy domestic sheep, they most frequently culture Mannheimia hemolytica. And the B. trehalose and the Mannheimia hemolytica that they culture usually has high leukotoxin activity. Doesn't bother the bighorn sheep. They deal with it when they're healthy just fine. They're carrying it, no problem. So when they are looking at another little thing that these bacteria are doing is they're working behind the scenes to confuse us. And this is some interesting, if anyone, I've got copies of all these papers. If anyone's interested in reading these, it's really fascinating what they're beginning to find out about these bacteria. They know that when they take Mannheimia hemolytica and put it into bighorn sheep, it kills them. But when they go up and get these dead sheep off the mountain and bring them in, when they culture their lungs, most of them they're not finding the Mannheimia hemolytica like you think they would. Why are they finding the Trehalose and why are they finding the Maltosida when we know it's the Mannheimia hemolytica bad guy that gets in there and kills them? So they started looking at the little interaction between these bacteria. And what they found out is that Trehalose, and this is when doing studies in the lab on a culture plate, when they get in there, the Trehalose grows faster, and basically it gets in there and outcompetes everybody else, especially the Mannheimia hemolytica. So if they check those after 6 hours, if they smear out a big goo of dead sheep lung, and they look at it after 6 hours, they can't even find the Mannheimia hemolytica, even though when they checked it at 4 hours, it was there. By the time they look at it at 6, it's gone. So what they think, here are some of the things that they think are happening. They think that when we are going up and dealing with the die-off, if an animal dies, the bacteria, once the heart stops beating, those bacteria don't just go, oh, we're dying. they continue to grow. So what may be happening is we go up, pick up a dead sheep, by the time we get some of those lungs out onto a culture plate, the Trehalose might have already outgrown and outcompeted the Mannheimia, and that may be one of the reasons why they're not finding that bad guy Mannheimia when they look at these. The other thing that they think is happening is that perhaps the Mannheimia has a different role to play. It goes in, it usually has a lot of leukotoxins. It goes in, lures all the white blood cells in, kills them all, causes a lot of tissue damage, and actually its job is done. It's basically done its thing. Allows some of these other bacteria to come in and kind of play around, and then eventually the animal dies. So they think that these two types of bacteria may actually have different roles. This is not only in bighorn sheep, this is also in cattle and domestic sheep. Same bacteria playing the same kind of little dirty tricks, but it's confusing to us when we're looking at it. So there was because of all these things where they could never find the kind of smoking gun bacteria, there's been a lot of question over the years, very, very contentious camps about whether domestic sheep can transmit disease to bighorn sheep. So there's a researcher up at the University of Washington who's been supported by all of the non-for-profit bighorn groups, have all chipped in money, is being supported by the university up there, and they just completed a study where they've basically proven that Mannheimia hemolytica can be transmitted to bighorn sheep. So what they did was they took the little domestic sheep they took Mannheimia hemolytica out of them that they knew was bad, had the toxin, and they labeled it with a green fluorescent label that they could figure out doing their tests. Then they went over and they have a group of captive raised bighorn sheep, and they tested all of them, and they did not have this Mannheimia hemolytica with this leukotoxin. They can identify them down to that level. So they have them in their two separate pens, 10 meters apart, for 30 days. Then they went ahead and they moved them. They moved the bighorn sheep next to the domestic sheep. They weren't in with them, but they could run back and forth along the fence line and do whatever they want. At 60 days, near the end of 60 days, they caught up all the bighorn sheep and found that 3 out of the 4 of them now had this little green fluorescent marker. So they didn't have it to begin with, so the only way they could have gotten it was to have it come across from the domestic sheep. So they knew that it had to come from there. Then they went ahead and commingled them, and in 4 to 9 days, all of the bighorn sheep had died. I think they euthanized one of them that was sick, but 3 of them died. And when they went in and cultured out the lungs and looked at the bacteria, they found that all of the Mannheimia, that there was the presence of this Mannheimia hemolytica in the lungs that had been tagged with this green fluorescent marker. So that proves, once and for all, in my mind at least, that yes, we now know that it can happen. Something that the animals were telling us all along, we now know can happen. So that's the story with the Pasturella family. Now, unfortunately, they're not the only ones that play in the game. And what we found, certainly in this die-off, is that we've had another player come in that's gotten involved with this pneumonia outbreak. And they're finding that also in around the West. There's something called Mycoplasma ovipneumoniae, which means sheep pneumonia. And there's a cattle mycoplasma and a swine mycoplasma and a human and a bird. So everybody's got their own mycoplasma. For many, many years, the domestic sheep folks have known that mycoplasma, when it gets in lambs, can cause kind of a low-grade chronic pneumonia, but it doesn't kill them like we know the Pasturella does. So we know that mycoplasma can cause, that's how it got its name, can cause these low-grade pneumonias, but we felt very certain in most domestic species that it does not go in and kill the animal. And the way it does this is actually, you're looking at scanning electron micrograph of your trachea. And you know how you have all those little cilia in there and they brush up the mucus to keep all the bad bugs out? Well, what the mycoplasma does is it kind of paralyzes the little cilia. So all the bacteria that live normally in the back of our throats get to crawl down, and the little defense mechanisms that's brushing them back up aren't working. So the bad bacteria and all their friends can all go down in the lungs and party, which is a nicer, warmer, wetter environment for them to be in. The other thing they found, again, that's confusing is M. ovinemoniae doesn't just come as one single bug. There's all sorts of different strains. And when they've looked at this in domestic sheep, they've found one sheep can have numerous strains of mycoplasma in it. Not all mycoplasma are created equal. Some tend to be nastier than others. But again, the general feeling on this is that it does not kill the animal. It just causes kind of a low-grade pneumonia. What it does is set the stage for the other players, the bad Pasteurella, to come in and kind of kill the animal. So what they did in some other experiments was they took M. ovinemoniae and they isolated it out of a domestic sheep. They inoculated, and by inoculating, I mean they're like spraying it in their tracheas, so they're really giving it to them, and put it in the bighorn sheep. And then the bighorn sheep kind of developed mild respiratory disease. They got fevers, they coughed, but they might have been a little bit slow for a few days, but then they recovered from that all by themselves. However, they were still carrying the mycoplasma. Then when they took the M. ovinemoniae and mixed it with the bad Mannheimia hemolytica and put it in the bighorn sheep, they were dead in 48 hours. So they killed them pretty quickly. So we know that the M. ovinemoniae can set up these sheep for the other bacteria to move in. They also went back and looked at some of these herds around the West that have died and had pneumonia outbreaks, because there's been many of them, and they went back and looked at them serologically. When they looked back at bighorn sheep herds that had not ever had a pneumonia outbreak, they found that they did not have, even though they had Pasteurella and had some Mannheimia living in their respiratory tracts, they did not have any titers to mycoplasma. So they were mycoplasma free. Then when they also looked at animals that were apparently healthy but had been from herds that had experienced die-offs, 60% of those animals were seropositive for mycoplasma. So even though animals that had die-offs and the animals had seemed to recover, they still found that some of those animals had been exposed to mycoplasma in the past. So certainly there's other players that have been involved and talked about in bighorn sheep pneumonia. Some of the infectious agents, there's respiratory viruses out there that cause shipping fever pneumonia in cattle, and also can affect sheep and goats, domestic sheep and goats. And there's been some PI3, BRSV, IBR, are all common viruses that are found that attack the respiratory tract in ruminants. And there's been some believed to have been some mild pneumonias and some die-offs with some sheep in Colorado where they found some PI3. And certainly when we look across the board at a lot of our bighorn sheep, we know that they've been exposed to these viruses. What we can't find is any evidence of actually the virus living in the lung tissue. So in this particular die-off, we don't feel that those viruses actually played a role because we weren't able to get them out of the lung. Parasites, lungworms has often been implicated, and certainly in young lambs, they will have high parasite lung worm loads. We found one animal that had some lungworms, but by and large, looking at the lungs on the rest of the animals, we did not find any significant or important amounts of lungworms in this. And then Pasteurella common to other species, again, there's been some studies that have looked at Pasteurella in other ruminants that potentially can cause some problems in bighorn sheep. What other non-infectious agents might be indicated? Trace mineral deficiencies have certainly been talked about a lot. We know that all of the soils around the West are marginal to deficient in selenium. The East Humboldt herd has had some selenium supplementation. We've had some blocks out there. Most of the animals were tested for blood or liver levels of trace minerals. We have not seen at this point that they have screamingly low levels. They have low normal to some deficiency levels. The at this moment in time, we're not sure exactly what role that plays. Naive animals, again, when you mix, that's why shipping fever pneumonia, when people are shipping cattle and hogs around the country, that's why there's such a huge industry in these antibiotics, is because when you take a group of animals from here and a group of animals from here and a group of animals from here and a group of animals in here, mix them all together in a feedlot or a truck, that's stressful. Some of these animals have never seen these bacteria that these other animals have, so those animals are naive and they are much more susceptible to whatever these guys have, and vice versa, because they just have never been exposed to it. And when we are exposed to things, we can build up immunity, and certain sheep and goats and cows can build up some immunity levels to some of these bacteria. If they've never seen it before, they have a hard time doing it. And again, what other environmental stressors? Certainly low food, crowding, those types of things that are players in all forms of pneumonia, in whether you're talking, again, bighorn sheep, cattle, or domestic sheep. Some of the things that's been brought up, it's like, okay, well, does everything have to have nose-to-nose transmission? When these sheep get together, do they all go down the line and kiss each other? They did some studies looking at the transmission of Pasteurella. They did find that it can, in certain conditions, it likes it things cool and wet and moist, that some of these Pasteurella bacteria can remain viable when they were blown through a wind tunnel for up to 60 feet. So they were able, and again, this was culturing them off a culture plate, but they were able to grow them out. So we know that if an animal's sick and coughing and sneezing, that might be a good way to transmit bacteria. They've been able to culture some of it off the grass. If an animal has a snotty nose or sneezes, blows a big booger on the grass and another moves on, that can live for a while depending on the conditions. In some of their studies that they did, they cultured bacteria off of straw, again, probably looking at animals that are going to fairs and being trucked, that type of thing. They found that at 24 hours at 77 degrees, the Pasteurella, you could still grow it. And then again, it likes it a little bit cooler and a little wetter, so at 40 degrees, it was able to survive for 48 hours. The water one is a little bit hard for me to interpret because, again, that was stored water. It probably wasn't put out in the middle of the sun, but I'm sure that it can survive in water for a certain amount of time, again, depending on the conditions. Mycoplasma ovipneumoniae is not nearly as tough. That definitely could probably survive being sneezed into somebody else's face or coughed into somebody else's face, or if you were all grazing around the same area and nibbling on something tender, but it's much more fastidious and it's not going to hang around in the environment. It really needs to be in the host. So what did we find? What have we found out so far from what we've done with looking at Nevada's animals? When we cultured them from both pharyngeal swabs that we took on live animals and also lung cultures that we took on dead animals, we found what we thought we would. We found a lot of Pasteurella trellosi. We found a tiny bit of Pasteurella multocida, and we also found a tiny bit of Mannheimia hemolytica, especially on the lung cultures. What they did is this lab also went back and they looked for the presence of this Mannheimia, because they know that it gets out-competed when it's on the culture plate. We went back and looked for the evidence of it, the evidence of its DNA. So they went back and did this PCR, which basically looks to see if it was there or not, if it left some of its DNA. So they went back, and on the 100% of the ones where it was not present in culture, they couldn't culture it off the lung. When they went back and looked at it, if there was evidence of it with DNA, they did find out that the Mannheimia hemolytica was there. So we know that it was there, went in and did some bad things. The interesting thing that we found is the fact that when they did culture the Mannheimia hemolytic and look at it, when it was present there, but the toxin level was low. So it wasn't really one of the ones that kills them in 48 hours. And that actually fits very well with our die-off. This started in late December, and we're still losing animals now. We had animals that we know, because they were radio-collared, were sick for weeks. This is not something that went in and boom, within a week we had 100 animals dead on the ground. So we know that we have the leukotoxin production And the Mannheimia hemolytica that's in there is not a real nasty one. We know we have not so nasty one. It's still killing them, but it's not doing it very quickly. So the other thing that we found, and we've gone in and we have cultured in the pharyngeal area, the nasal, the sinus, and the eardrum, and the lung, is we found Mycoplasma ovipneumoniae everywhere. So we found it on 100% of the animals that we've tested, we found it. We also went back and did, on live animals that we had, we went ahead and did the serological titers. We found pretty high titers to this respiratory virus, PI3, but again, we've kind of gone back and tried to look for it in the lung to see if it was actually down there growing, and it does not appear to be there. So they've been exposed to it, which is not uncommon, but it did not seem to play a role in this. We've also gone back and looked at serological titers and seen that, yes, we do, in fact, have some exposure to Mycoplasma ovipneumoniae. Again, we looked at parasites for all of these animals. The Eimeria nematodyrus and protostrom are two intestinal parasites, and again, these animals were not carrying huge burdens of parasites, which we wouldn't expect in rocky areas of Nevada anyway, because it's dry and harsh, and parasites aren't that happy with that. And then again, the lungworms were not a real big problem. Trace mineral analysis, when we've done liver and whole blood analysis, it's low normal to deficient. It's not screamingly low. We're not fully understanding at this point. We're still doing some more forage sampling, looking at the availability of the selenium in the forage and getting an idea of how that compares to other sheep around that we sampled this winter, and looking at the forage they ate, so we can kind of compare their selenium levels to what they were eating that didn't have a die-off with selenium levels in the rubies and the East Humboldts that did have a die-off. Let's go back and look a little bit at our complicating factors. I think that we understand now why they're not getting a single pathogenic species, is that these bacteria that are getting in there are playing their own little host of games when they're in the animal. Why do some bighorn sheep and domestic sheep encounters do not always result in a die-off? Well, I don't think that all domestic sheep are probably not all carrying leukotoxin, bad leukotoxin genes, and some of these bighorn sheep may be developing immunities to them. So I think it's probably a combination of factors. If there's a real bad one in there that's got a nasty leukotoxin, then they may be able to kill a group of bighorn sheep. Obviously, that wasn't really the case in this die-off because they didn't die really quickly. And they can adapt and develop some immunities to some of the shared bacteria that they have. Bighorn sheep can experience a die-off with no known prior contact with domestic sheep. I think that every ruminant species can always have some sort of pneumonia die-off, and certainly bighorn sheep can have extenuating circumstances that are not related to domestic sheep that can lead to outbreaks of pneumonia, just like domestic sheep who seem to live very, very well with highly toxic bacteria in their respiratory tract. Something can trigger that, and they can get pneumonia and die too. And we know that we did culture Mannheimia hemolytica from a number of these sheep, and it had fairly low toxin levels. So again, we know we can get this bad guy out of there, but in our sheep, in this particular group, for this particular die-off, they had fairly low leukotoxin levels. So some of the things, this is something interesting that Nevada came up with all on our own, is that when we kind of discovered that we had this mycoplasma, that mycoplasma likes to hang out in the sinuses and the eardrums. And this is usually what we find with die-offs. This year, our biologists and sportsmen were out on the mountains helping us. We wanted to get as many samples as we could, and they were devotedly out there picking up carcasses and trying to get us lung tissue and head tissue. But in many die-offs, all we'll find is something like this, and many times nothing even this good looking. Sorry for the grossness of the picture, but it'll wake everybody up. This is the front of a bighorn sheep that we cut through we basically cut the front of its face off, and you're looking back toward the back of it at its sinuses. And if you've ever had bad sinus headaches, I imagine you probably think your sinuses look like this. But this is all sorts of blood and thickened tissue and hemorrhage. Even over on this side, it's still pretty red and irritated looking. Go back. But what we did is we were able to go back and culture some of these areas, culture some of this tissue. All right, well, never mind. And find out that they were carrying Mycoplasma pneumoniae, which we know now has predisposed some sheep to some of these respiratory infections. So this is a tool that I think will be able to help us now in one sort of surveying what we've got going on in maybe hunter-harvested sheep, and also going back if in a die-off, if we've been able to pull some heads out of freezers that were involved in previous die-offs, and at least be able to realize that we had Mycoplasma pneumoniae as a player in that particular situation. So that's something that we started looking at, and now they've gone back in some of the other states and dragged back these gnarly, dried up heads and have been able to find a fair amount of mycoplasma out of it. So it's an interesting thing that we can use as a tool as we go forward. And that's all I have.
There will be a test.
That's right.
Uh-oh.
Where's the test?
Sorry, I was thinking of naming all the little bacteria and they could be little hand puppets, but I don't know if...
That was very interesting. I had a couple questions.
Yes.
It looks like you're getting a long way with the bacteria, identifying the causative agent here. Is there any work going on with, autogenous vaccines or something now because we got a situation where someday it looks like this stuff's in the grass, so we're not going to ever defeat the bacteria. So the only way we're going to genetically get some sheep that are resistant to this before we transplant sheep into this, maybe inoculate with some type of vaccine against these or something like that?
I think they have played around a little bit with looking at there are some Pasteurella vaccines that are made for cattle. They don't do all that well in domestic sheep because pneumonia is probably one of the biggest killers of domestic sheep. So then they've tried doing it in some bighorn sheep. They worked on it a little bit in Colorado, but it's never really taken off. One of the, Dr. Shree, who's the professor that's the FNAS endowed chair working on bighorn sheep pneumonia issues at Washington, his hope is that he would like to see bighorn sheep and domestic sheep be compatible on the landscape, with obviously an easier time looking at domestic sheep, making them, because they're easier to get to and you can manipulate them a little bit more easily. One of the things that he's very interested in is looking at the Pasteurella trehalosii, which seems to be an aggressive competitor, and potentially taking one of those that has low leukotoxin activity and maybe using that as an inoculant in domestic sheep to get rid of the Mannheimia. Now, that science is in its infancy right now, but that's one of the things that he is interested in. We did try using an antibiotic called Draxin, which is used for shipping fever pneumonia in cattle and swine, and we talked to Pfizer, who's the company that makes it. We don't really feel that we got amazing, turn your head results during this outbreak, but I did talk to them. Their swine group, they have a swine unit because it's licensed for swine, and Mycoplasma suis, or whatever, I can't remember what it's called, causes a lot of low-grade chronic respiratory disease in swine. And their swine group, in very, very controlled situations, was trying to use Draxin to eliminate Mycoplasma in swine. And last time I talked to them, their researchers who do this for a living were banging their heads against the wall. So they're now beginning to back off their thoughts that they can use Draxin as a way to eliminate some of these bacteria. So we were hoping that they'd want to jump on the bandwagon, but they're so discouraged trying to get that accomplished in cattle and swine that they're not, that doesn't seem to be a viable option either.
Thank you, Mr. I think I heard you say it, but now domestic sheep are known as the only carrier of these, well, let's just call it the specific bacteria that you're worried about. Other animals out there are not passive carriers of it.
No, they are all ruminant species. So deer, cattle, sheep, goats, all carry. If you
All three.
swab all
bad ones.
of these ruminant species, probably if you go to Africa and swab antelope, pronghorn antelope, all of them have Pastorella species in their pharyngeal area. Because we all have something in there, and we all have these commensal bacteria. What they found was, and bighorn sheep do, what they found was that these different, the Pastorella trahelosi, the Mannheimia, are the two that create these leukotoxins. Pastorella multocida, has anyone here ever had rabbits? Ever have big abscesses on the rabbits or in their ears? Pastorella, big problem with rabbits. That doesn't produce the leukotoxins. So these two, the trahelosi and the Mannheimia, create these leukotoxins. What they found when they went in and just cultured domestic sheep versus bighorn sheep that had never ever been exposed historically to domestics is that the ratio of the bacteria in their pharyngeal areas was different, and the amount and the types and levels of leukotoxins that these bacteria expressed were different. So domestic sheep can live very comfortably with Mannheimia hemolytica that has high leukotoxin activity, meaning it's very nasty. Doesn't bother them. It does, I'm sure, at some point, because when you stress them, ship them, that type of thing, it causes pneumonia in domestic sheep, which is bad. And when they look at, but it's very, very deadly to bighorn sheep, and it's because of the toxins, because the bighorn sheep neutrophils, when they get exposed to these toxins, they just implode and cause so much damage that the animals die very quickly.
So they're being carried by these different species out there. I'm just wondering, if you probably looked into it, now that they are out there, we're not having the deer carrying it around and spreading it, or the elk, if they're all carrying, they're not all carrying trahelosi, they're not all carrying Mannheimia hemolytica.
There are with the studies that they've done, when looking at the original Penn studies that they did, which kind of started this all off, is when they put bighorn sheep next to elk, they put bighorn sheep next to llamas, they put them next to cattle, they put them next to goats, they put them next to deer, and they put them next to sheep.
Okay.
And it was the ones that were next to the sheep. And
Okay,
that's where they sort of started looking, and because of the historical data, where they realized that when they mixed, and it would make the most sense because more related species are going to have easier time transmitting different similar bacteria. I'm sure that the bacteria in domestic sheep, those Pastorella, are more likely to infect bighorn sheep. Cattle Pastorella tend to be more specific to cattle. Their biotypes and serotypes are more specific to cattle than they are to domestic sheep. So even though they're the same family, they kind of tend to hang out in their own different species.
Thank you.
Yes. Oh, sorry.
Thank you, Mr. Chairman. Peri, meanwhile, we have 105 out of 180 die off in the East Humboldts. And the 34 figure that you showed on the thing there, I saw that a month ago. Have there been no die-offs since then, or do Do you anticipate there will be? Or have you just not seen more because of the winter conditions up there? Or what?
Mike here.
Well, I hear that tomorrow.
Those were the numbers that I got from Caleb, the biologist, as of the beginning of May. So we've definitely had lower numbers in the Rube East, but it's also much more difficult to get up in there and find sheep. That's probably one of the biggest issues.
I'm sorry, Mr. Chairman, I thought that was part of this agenda item.
Well, that's okay, we'll probably hear more about it when we have the quotas, you know. Commissioner Moore.
Thank you, Mr. Chairman. This is probably a difficult question for you to answer, maybe at this time, but I'm going to throw it out there. In your opinion on this die-off, from what you've shown us on artificial exposure, and I'm talking about artificial exposure or induced exposure, whatever you want to call it, you showed that when it was transmitted, all animals died. Like when you mixed the sheep,
Right.
all animals died. In this case, you have animals that are still alive.
Mm-hmm.
Okay.
Mm-hmm.
Do you think that it is because their genetic makeup has resistance to these bacteria, or do you think that they didn't get enough exposure to it to cause them to die?
I think that there is, and I'm trying to be as scientific as I possibly can, when they do the studies, they are, especially when they take a Mannheimia hemolytica with a known leukotoxin out of a domestic sheep, which is what they did, they have certain, and they test it to see how nasty it is. So they're not picking the ones that are not very nasty. They're picking the ones that they know are pretty nasty. So they know that before they went into that last study with the fluorescent labeled marker, they know that they had some Mannheimia that was more than likely going to kill those sheep. What we've seen in ours so far is our Mannheimia that they've cultured and tested the nastiness of it. It's not very nasty. So I think there's a couple of things going on. I think that ours wasn't going to kill. I mean, it killed a lot more in the East Humboldts, maybe because of the way they were clumped together there. But there are live sheep in the East Humboldts. Either they were hanging on the periphery, or they were genetically different, or they were able to mount an immune response, a better immune response, or they didn't get as high a dose, or they may not have mycoplasma. Because I think in this particular die-off, that the mycoplasma has played, it's that little group of them together that's played a big role. So I think we had a less nasty Mannheimia. We know we did because they looked at the leukotoxin and it was low. And that some of these sheep, are going to be survivors. And they are going to be genetically, I mean, do we know, can we say, oh, this ram doesn't get sick from pneumonia no matter what you throw at him, so hey, let's bring him into captivity and he's going to be our new breeder. May not be that cut and dried, but certainly there's going to be animals that develop a better immune response and stronger antibodies to fight it off, or for whatever reason. I mean, just like if you inoculated everybody in this room, a couple would be dead by the end of the day, some would linger for a week, and a couple would, be okay. And a lot of other factors that we don't understand. We're going to draw straws, figure out
I was
who's.
going to say the board first, commission first. We're willing to see you come back tomorrow.
Harry?
The calves first.
It seems like,
me.
the surviving sheep, those are the ones you should be studying, maybe for a vaccine, because like in humans, you get Haemophilus or pneumococcus, and we've got a vaccine for elderly people for pneumonia. And, so the ones you want to study are the ones that are survival, because there might be something in there that they have some type of, develop some type of immunity over a period of time. Viruses are hard to culture anyway.
Right. And part of our study that we are, our ongoing study, and one of the main reasons that we really wanted to radio collar a bunch of sheep, we got about 30 animals that we had originally radio collared, was so that we could go back and find those specific animals that we knew had survived the outbreak, and we have or sick cultures on them. We've got sick blood that was taken while they were sick, and what we're most interested is what they look like, down not only 6 months, but 18 months, 24 months, 36 months down the road, and also follow up on lambs that are born to see, because one of the issues that everybody's concerned about, and we didn't see this the last die-off, we've had very good recovery in both the East Humboldts and the Ruby Mountains since the 1996-97 die-off. We were lucky. We had our populations come back. We want to check this year, or this time, and see, are we getting these lambs that are dying at 1 to 3 months of age? And what they think is happening there is that the lambs are with their moms, and then at 1 to 3 months of age, they start getting together in these little nursery groups. And when you get all the little kids from all these neighborhoods and send them to nursery school, somebody brings in the snotty nose, and pretty soon everybody's got it. So that's the theory on that, is that two little sick lambs get there because they got something from their moms and kill the whole group. So we want to be able to see who gets born and how long they live and what happens with them. And yes, the research community is very interested in what is actually in the lungs of these animals that do survive.
Okay. Commissioner Capurro?
One last question. I've talked to hunters who had taken sheep off in this case, it was the North Rubies, 3 to 4 years after the 96 die-off. And when they opened them up, they still had the black lung. So even
Right.
though they appeared healthy,
Right.
they really weren't.
Mm-hmm.
So the question becomes, do any of them regain their health, or is it simply they're living with the disability that they have?
And I think probably there is, some of both. I think the ones that are really severely infected never, that once that lung is adhered to the side of your chest, it's not going to unadhere, no matter how much time passes. And some of that lung will just never really recover. I think one of the questions is, are those animals shedding something, or are they, in essence, just have a walled-off infection, and what's coming out of their nose is nothing that's bad? And then that there are probably some animals that never got that sick to begin with, and they hopefully made a full recovery. But those are questions that, why it's so important for us to continue to do the follow-up work, is to be able to hopefully start putting some of these pieces of the puzzle together.
I don't think those sheep that have been infected years ago, just like in humans, you get tuberculosis, the body encapsulates it with calcium, and that lung tissue is destroyed.
Yep.
And when your body gets in a weakened condition and absorbs the calcium, the TB breaks out again. So I think you're always going to see that diseased lung. It's not going to heal. It gets encapsulated or something, the animal puts up with it, and some of the ones that were taken from the last, 10 years ago, they had, running on 50 or 70, they were losing 50 or 75% of their lung capacity,
Right.
and they were still alive. It encapsulated, destroyed the tissue, and the lungs, you don't need that much lung to survive. A little
Right.
piece of lung will keep you alive for a while. And I think that's what you're seeing. So those sheep that are surviving now are permanently damaged. The ones that are surviving, they have reduced lung capacity, I think, and if you went to look at them, if they were infected, I think they're all going to be reduced capacity and lung capacity. If you could, 10 years, we'll find that out. Okay, thank you, Peri. Very, very good
Chairman.
presentation.
I'd like to make one observation. I think one of the things that Peri did a good job, there's been an argument over the years, does domestics give Bighorn, the disease. I think that's coming out in the journal. That's pretty well decided. And I'm looking forward to the time we can get beyond that discussion and that argument.
Yeah.
Now let's figure out what we're going to do from a management perspective, because there's a number of things we can do. We need to be creative there. Number one. Number two, that in no time have we pointed the finger at the livestock industry, the sheep industry, and said that's the cause of our die-off. We simply don't know, and we've made it very clear in our news releases. Made it very clear that we're not pointing the finger at the livestock industry whatsoever. And I think, thirdly, when we move on from here, I think, and Mike will tell everyone that all the easy mountain ranges are pretty well finished up. We're going to be looking at releasing sheep in places where they potentially could rub noses with domestics. And I think that we need to work together on a way that we can hold the livestock industry harmless, that they're not worried about sheep die-off. That will be on us if we decide to do that. So I think we're planning some things for the future, and hopefully maybe we can inoculate the domestics and we won't have this problem. That's nirvana, quite frankly. So anyway, I thought I'd leave with those three ideas.
No, you're absolutely right. I don't think the department's ever accused them of that. And I think the sheep people would, they don't mind as long as they're hold harmless.
Yeah.
And there might be a way we can intermingle them, these populations, and we have to get a super breed, or something like that. But I think that's absolutely where we have to go in this state. Okay, let's move on to the next item here.